Hello everyone, hope everyone is safe and sound and staying at home. I'm Arita and I'm once again back on a study with a biology lesson. And today is the second lesson on enzymes. So without any further delay, let's just get started. Today, we will be starting off with environmental effects on enzyme functions. So active sites or enzymes are very sensitive. They sense even the slightest change in the environment and respond accordingly. So some of the factors that affect the active site and subsequently enzyme function include temperature. So temperature affects the rate of enzyme reactions. Enzymes have an optimum working temperature often, but not always close to that at which they usually function. So now what do we mean by an optimum temperature? So each enzyme has a temperature range in which a maximal rate of reaction is achieved. So this maximum is known as the optimum temperature of an enzyme. So the suitable temperature for enzymes to function properly is 37 degrees Celsius. Increasing or decreasing the temperature above 37 degrees Celsius affects chemical bonds in the active site, making them very less suited to bind with substrates. And higher temperatures denature enzymes. So by denaturing, what we mean is taking away the natural qualities of something or destroying the active site so that the substrates cannot bind with the enzymes anymore. So that was it about temperature. Now, how does the pH affect the enzymes? So enzymes are affected by the acidity or alkalinity of the solutions in which they act. Some work best in acidic solutions and others require slightly alkaline solutions. So what happens is amino acids present in the active site are acidic or basic. And fluctuation in pH can affect these amino acids, making it hard, really hard for the substrates to bind. And extreme pH values can denature enzymes as well. So now, let's talk about enzyme concentration and substrate concentration. So increasing enzyme concentration will increase the rate of reaction as more enzymes will be available to bind with substrates. However, after a certain concentration, any increase will have no effect on the rate of reaction. The same thing goes for substrate concentration. Increasing substrate concentration will increase the rate of reaction, and this is because there will be more substrate molecules to be colliding with more enzyme molecules, and thus more products will be formed. But then again, this effect is valid only up to a certain concentration. Now, we will be talking about the inhibition of enzyme activity. So some really evil substances called inhibitors reduce or even stop the activity of enzymes in biochemical reactions. They either block or distort the active site, thus inhibiting the reaction. Based on this, there are two types of inhibitors. So the first type is the competitive inhibitor. So what the competitive inhibitors do is occupy the active site and prevent our substrate molecules from binding to the enzyme. And the second one is the non-competitive inhibitor. So what they do is they attach to parts of the enzyme other than the active site. Like they would attach to all the parts except for the active site, which is the main one, to distort the shape of an enzyme. So they are really evil, aren't they? And lastly, we will be talking about how the enzyme-catalyzed reactions are reversible. As in the previous video, I have discussed already about the two models, the lock and key hypothesis and the induced fist hypothesis. They are all reversible. So enzyme-catalyzed reactions are mostly reversible. So here, as you can see, the first step shows the substrate entering the active site of the enzyme. So the enzyme changes slightly as the substrate binds and enzyme substrate complex is formed. So now the enzyme or product complex is there, and then again the products leave the active site. So they are reversible. Thank you so much everyone for watching today's video. I really hope you enjoyed it, and hopefully we will meet again in another video. So have a good day. Thank you.